Hello everyone, my name is Liu Shangming. Nice to meet you in MOOC. Today, I'd like to give you a lecture about twins. In everyday life, you may encounter twins. Do you know how twins produced? Do you know the differences between fraternal and identical twins? Then, let's begin. In most pregnancies, a single embryo develops in the uterus, but sometimes it can be twins. That means two embryos develop together. In this picture, it shows two embryos connect to one placenta. And in this picture, it shows two embryos develop together in the uterus.
There are two kinds of twins, dizygotic twins and monozygotic twins. Most twins are dizygotic, also known as fraternal twins, meaning that it originates from two separate eggs fertilized individually. Another type is monozygotic twins, or identical twins, which come from a single zygote that quickly split into two separate groups of cells. Most times, dizygotic twins happen when the ovary releases two eggs simultaneously. Two different eggs are fertilized by two different spermatozoa, so they have completely different genetic makeups. They may be of the same sex or different sexes. External and genetic features are no more alike than regular siblings. The only thing they have in common is that they were in their mother's uterus at the same time. Dizygotic twins always have two amniotic cavity, two chorionic cavity, and two placentas. Sometimes, however, the two placenta are so close together that they fuse. Similarly, the wall of the chorionic sac may also come into close opposition and fuse. These two babies, one girl and one boy, they are fraternal twins. Fraternal twinning shows a hereditary tendency. Monozygotic twins come from a single zygote, splitting into two embryos. They have the identical genetic materials, so they have the identical blood groups, sex, and external appearance. These two babies are quite same. They are monozygotic twins. For monozygotic twins, the splitting of a single zygote happened at various stages of development. The earliest separation occurred at the two-cell stage. Each embryo has its own placenta, amniotic cavity, and chorionic cavity. Splitting of the zygote usually occurred at the early blastocele stage. The inner cell mass split into two separate groups of cells within the same blastocystic cavity. The two embryos have a common placenta and a common chorionic cavity, but separate amniotic cavity. In rare cases, the separation occurred at the bilaminar germ disc stage, just before the appearance of primitive streak. The two embryos share one placenta, one amniotic cavity, and one chorionic cavity. Sometimes, such twins may not fully separate, resulting in conjoined identical twins. These two babies joined from front to front, called serocopgas. These two babies joined bottom to bottom, called pagopgas. Joined side to side, also known as bicephalus, joined head-to-head, also called craniopagus. Some conjoined twins are connected to each other by skin or cutaneous tissue. Such twins can be successfully separated by surgical procedure. However, if they have a vital part in common, anatomical relations in some conjoined twins do not permit surgical separation.
Okay, this class is over. Now let's record what we had learned in this class. First, we learned two kinds of twins, fraternal twins and identical twins. Then we watched some pictures about conjoined identical twins. Now that's an interesting case for you. An ultrasound examination reveals a twin pregnancy with a single placenta. Chorionic villus sampling and chromosome analysis reveal that the twins were likely female. But at birth, the twins were of different sexes. Do you know how could this error have occurred?